Hello everyone and welcome back. This is the second lecture in a series of lectures on the cellular level of organization. As you recall, the last time we were together we had described the basic structure of a cell, the various parts of that cell. We had gone into some detail about the plasma membrane. We described what kind of molecules the membrane was composed of, how they were organized. We had talked about proteins and all the functions of the proteins. So now we're going to put that knowledge to use and talk about this process here, transport across the plasma membrane. Why is this subject important? The answer to that question is that your body cells, in order to be healthy, they need certain things. And they need to get rid of certain things. So they need to get rid of waste products, carbon dioxide, hydrogen ions, a variety of various things that accumulate in the cell as a result of normal metabolic activities. They also need things like oxygen, glucose, amino acids. So for cells to be happy, there needs to be this constant exchange between the outside world and the inside world of the cell. And that exchange has to take place over the plasma membrane. So what we're going to look at is those processes that allow that to happen. So that's why this is labeled transport across the plasma membrane. As we look at these processes, we can divide them into two major groups. So there are two major ways that transport happens. Number one is active transport. Okay. Active meaning that the cell is active in supplying energy for these processes to take place. So the cell will use ATP. It is going to require the cell to expend energy. It's not free. The cell needs to expend energy. And this is going to, this is the result of, I should say, it's a result of that the movement is going to be against the concentration gradient of whatever is being moved. So against the concentration gradient. The cell uses energy in the form of ATP, and the movement is against the concentration gradient. That is the hallmark of active transport. The second form in which this takes place is passive transport. So passive transport, as you might have guessed, requires no energy on the part of the cell. No ATP is used. Okay? And as you recall, ATP is the energy currency of that cell. Okay? So no use of energy on the part of the cell, which is different than saying that there is no energy in this system. There is energy, otherwise it would not happen. But the cell does not have to supply that energy. And movement in this case is down the concentration gradient. Moving down the concentration gradient. Okay. So, when we look at all of these, we can identify three passive processes. There are three passive processes. Number one, simple diffusion. Number two, facilitated diffusion. And under facilitated diffusion, there are two subcategories. They are channel-mediated facilitated diffusion and typically, though not necessarily always, what we're talking about here are ion channels. The vast majority of things that are moving through this mechanism are in fact ions, and they're moving through what we call ion channels. We'll get into that a little bit more later on. So channel-mediated facilitated diffusion is one form of facilitated diffusion. The second is carrier-mediated facilitated diffusion. Here we're going to use a transport protein. And if you recall in our discussion, transport proteins are working to move something across the membrane as a result of the change in shape of that protein. So what we're going to get is a change in shape of the protein. That is carrier-mediated facilitated diffusion. In the case of the channel-mediated, there's no change in the shape. It's simply a doorway, a channel, if you will, through the membrane, through which certain very specific molecules can pass through, others are prevented. This is different because the protein changes shape as the molecule moves across the membrane. So that's two, and the third process is osmosis. And osmosis is the movement of water as opposed to the movement of solutes. So the other two that we had talked about were movements of solutes, such as ions or glucose or amino acids. Here we're talking about the movement of the solvent, and in our case that's going to be water, so the movement of water. And the mechanism of movement across the membrane, or perhaps a better way to say this, the pathway of movement is in two ways. Number one, between the phospholipids that make up most of the membrane, if you recall phospholipids. So in between those phospholipids, water can slip into or across the membrane. And the second one is through special proteins. And these are relatively new. We did not know about these for a long time. So these special proteins have a name, and they are referred to as aquaporins. Aqua referring to water. There are a variety of different aquaporins. We're finding new ones all the time. So this is kind of an interesting story because, as I said, for a long time we were totally unaware of the presence of these proteins, and we simply thought that water was moving into or out of a cell by sneaking through, or between, I should say, the phospholipids. Well, it turns out that's not entirely true. And the way we discovered these aquaporins, is when we were looking at the kidneys, we noticed that there were a certain group of cells which apparently can control the movement of water across their membrane. So sometimes water would move, sometimes they didn't, depending on the needs of your body. So for example, if you were dehydrated, less water would move across the cell membranes of these particular cells and you would produce less urine. If you were overhydrated, just the opposite would happen. So it was kind of curious that these cells seemed to be controlling the movement of water, and so that led to an investigation and then the discovery of these proteins. Okay. All right, so those are all three ways in which passive movement occurs across the cell membrane. There are also three active transport processes. These are primary active transport, secondary active transport, and third is vascular transport. So those are the three active ways in which processes or substances cross the membrane. Now, under vascular transport, we can break this down further into endocytosis into the cell, exocytosis out of the cell, and the third form is phagocytosis. And phago means to eat or to consume, and that's a special form of vascular transport which you'll become much more familiar with when you do the immune system, and I will talk briefly about that towards the end of this lecture. All right. So those are the mechanisms or the pathways in which things can move back and forth across the membrane. Let's take a look at some of the principles that will guide that movement and some of the natural or physical laws that are involved with the movement of substances across that membrane. Okay. So let's go ahead and we will title this portion of our notes Principles that govern membrane permeability. Okay, and transport. All right, so let's go ahead and begin with the first principle, and that's going to be diffusion. So let's get a good working definition of what diffusion is. So this is going to be the random mixing of particles in a volume of space. As a result of the kinetic energy of the particles. Woohoo. So that word kinetic is an important word to pay attention to. And let's go ahead and quickly draw a couple of scenarios to talk about talk about diffusion, and in particular this idea of kinetic energy and how that relates to the movement of substances. So what I'm going to do is I'm going to just simply draw a space. I'm going to define a space, and that's our space, because you notice that it talks about a volume of space. So we're defining this little box as our volume of space. And we're going to go ahead and we're going to put in some particles into that space. Well, there are the particles. And what's going to happen is that these particles will have kinetic energy. So what is kinetic energy? Kinetic energy is the movement inherent in a mass. And that's it. So it's the movement of a substance. So everything around you has kinetic energy. So that is in the air. So all the oxygen molecules, the carbon dioxide, nitrogen, all those molecules are zipping about in the atmosphere. Now, you don't feel that for a number of reasons. First of all, they're very small, but also they're hitting your body in different directions all at the same time, so you don't feel any pressure of that kinetic energy. But all systems contain kinetic energy unless you're absolute zero, and not Fahrenheit and not even Celsius, but Kelvin, absolute zero, and then there is no more kinetic energy, and we don't know what the state of matter would be in that condition. But be that as it may, what I've drawn here is a concentration of these particles in this corner. And because they have kinetic energy, they're moving. And they're moving in a straight line until they hit something. And what they're going to do is they're going to hit their neighbors. So that molecule is going to hit that one, and it's going to bounce off, and that one will bounce off. And they're just going to be hitting and bouncing off in a variety of different directions. And the ones out here at the outer edge, because there are none of these things to bounce off of, they will continue to travel in a straight line until they do hit something and then bounce off of that, and that, so forth and so on. And so basically what's going to happen as a result of the kinetic energy, there will be a tendency for these molecules to move out from this concentration and fill in that space in a random manner. Okay? So that is diffusion. And that happens in a gas, it happens in a liquid phase, it happens in many different types of ways. Now, one thing to keep in mind as we talk about this is that and I'm going to draw in some more here. And you notice that there are different particles. Okay, so they're different. And what the point I want to make here is that the same process will happen here. These pink particles will not see the orange particles. The orange particles are acting as if they are in the world all by themselves. They interact with themselves, but they do not interact with these. These will interact with themselves, but they will not interact with the orange particles. And so what will happen is these are moving away from this concentrated area to fill in that space in the same way that these are, but they're doing that independently of each other. And they will end up, in the end, if you let this system go long enough, what you will discover is that both of those will end up being evenly and randomly distributed in that space. This is important because let us consider that this would be oxygen and this is carbon dioxide. So we are now in the blood vessels and in the lungs, the blood vessels that are entering the lungs and in the cavity of the lungs. And so you want carbon dioxide to move out of the blood into the lungs, and you want oxygen to move out of the lungs into the blood. If these two were interfering with each other, that would be very difficult to happen because this arrangement would prevent the movement of these molecules, because this one would bump into that one and back and forth, and it would be hard to evenly distribute those in that space. And so because these are ignoring each other, oxygen will move from the lungs into the blood at the same time that carbon dioxide is moving out and into the lungs. Okay. All right, so that's diffusion. Another way to express diffusion, another way that we can write down the definition, and I'm going to do this, it's worth it, is the movement of substances from a region of high concentration to a region of low concentration until equilibrium is reached. It's basically the same definition. What we're emphasizing here that's a little bit different than the first definition I gave you is the reaching of equilibrium and the movement of high concentration, low concentration. But that's inherent with that other definition that we talked about. So they are not different from each other in the sense that they are defining two different processes. They're both the same processes. They're being defined, that same process is being defined in slightly different ways. Okay. All right, so that's diffusion. Now let's talk about some of the factors that are going to influence diffusion rate. So factors that influence the diffusion rate. Okay. So we know that diffusion is going to take place. It's going to move from high concentration to low concentration. But the question we're now asking ourselves is how fast is that going to happen? How fast will that particle or that substance fill in that space in an even and random manner? So there's a number of factors that are going to influence this, and we'll talk about each one of them, and I will try to apply each one of these factors to an example of your own biology. So the first one is the steepness of the concentration gradient. So the steeper the concentration gradient, the faster the diffusion rate. The steeper the concentration gradient, the faster the diffusion rate. Let me go ahead and give you two examples to illustrate what we're talking about. So I'm going to define two spaces, and I'm going to try to make them about the same size. So now the question is, let me first of all establish that concentration gradient. So here, that's that, and here, okay. So which one has the steeper concentration gradient? Okay. So this has a steeper concentration gradient. And the rate of diffusion is faster. Rate of diffusion here is slower. Simply because we have a large concentration, very concentrated here, nothing out here. Here, we do have a concentration gradient, but not, we do have these out here, so it's not quite as steep. So this is what we're talking about, the steepness of the concentration gradient. Okay, well, let's apply this to your body. Let's take two individuals. One is sitting down and watching television. The other one is running around the track, exercising. Okay, so the student who's just sitting there, the muscles in their legs are still using up oxygen. Even though they're not using them in terms of actual movement, there's still metabolic processes that require oxygen. So oxygen is diffusing into those cells. But it's not being used up very fast. On the other hand, the gentleman, so this would be more of this case here, that the person who's watching television. On the other hand, the person who's running, their leg muscles are exercising. They're producing ATP, and they are burning up oxygen at a tremendous rate. So oxygen does not last very long in the cell, and as a result, the concentration gradient of oxygen between the outside of the cell and the inside of the cell is quite large. And so oxygen will diffuse into those muscle cells at a faster rate for the person who's running than the person who is sitting down. So this idea of steepness of concentration gradient is working in your favor in that situation. Simply by burning up more oxygen, you create a steeper concentration gradient, and more oxygen is being dumped into your cells to feed the need for the production of energy. Okay. All right. So let's talk about a second factor that's going to influence the rate of diffusion, and that's going to be temperature. So we can say that the higher the temperature, the greater the kinetic energy. And the faster the rate of diffusion. Okay. So temperature is merely a measurement of the kinetic energy in a system. The higher the temperature, the more kinetic energy. The more kinetic energy, the faster those molecules are moving. Okay. So temperature is merely, as I said, a measurement of the amount of kinetic energy stored in a system. Okay? And so as temperature goes up, kinetic energy goes up, and the speed at which molecules move also increases, thus increasing the rate of diffusion. Right? So how does this apply to your body? Well, this is a factor which is not quite as important as some of the other factors. So, and the reason for that is that even when you're exercising and producing a lot of heat, we have already talked about this, your body is trying to maintain homeostasis, and one of the variables that it tries to maintain is core body temperature. And so it will start reacting to the increase in temperature by employing different mechanisms to lower the temperature, such as sweating, for example. And so internal temperatures don't vary that much inside the body. So this is not really that much of a factor. There is one area, although it's not directly related to diffusion, where this can make a difference, and that's when you have a fever. So a fever is an increase in the temperature of your core body temperature. And when that happens, the kinetic energy of all the molecules in your body, of course, are increased. They're moving faster. And many of the metabolic reactions that you're using then to defend yourself happen at a quicker rate. And so you're more able and more efficient to defend yourself from the pathogen. So temperature is something that you impose upon yourself for the most part, although it is true that there are certain toxic chemicals that are produced by bacteria which cause you to have a temperature. But temperature is, in a sense, as long as it's not too high, working in your favor by speeding up the chemical reactions which you're using to defend yourself. All right, that's temperature. Let's look at another factor, and this would be size. So size or mass of particles. And what we can say is that for any given temperature, or another way we can say that, energy, the larger the mass, the slower or the less kinetic energy the particle will have. And so all we're saying here is that for the same amount of energy, a larger object will move slower than a smaller object. Take, for example, if you were sitting there in front of two different balls. So one is a very small one, like a hard ball that's used in the game baseball, and maybe a very large one, bigger than even a basketball, something as big. So if you push both of them with the same force, the same exact force, the smaller object will move faster than the larger object because you have to overcome the mass of the larger object. So you put more energy into moving that larger mass than you do with the smaller one. As a result, for the same given amount of energy, smaller things move faster than larger things. And how does this affect your body? Well, again, coming back to the respiratory system, carbon dioxide is a much larger molecule than oxygen. So it's moving at any given temperature, which is your core body temperature, at a slower rate than oxygen. And so this is one of the things that you will take into consideration when you do the respiratory system. So how gases are exchanged in the lung are dependent on a number of factors, some of which we're talking about here. Other factors are also involved. But carbon dioxide, being larger, is moving at a slower rate than oxygen. And that changes the physiology of that particular gas relative to oxygen. Okay, so size or mass of the particles. Another factor is surface area. So the greater the surface area, over which diffusion happens, the faster the rate of diffusion. So surface area plays an important role in a lot of different areas of your body. And this is one of the reasons. So one of the things that you will find and see as you go through your studies is that there are certain areas in your body where the structure is greatly folded. So it's not just a smooth, flat surface, but it has these many folds in it. And when that happens, typically, although not always, typically that's a effort to increase surface area. And because you can get a more surface area within a defined space if you fold that structure inside that space as opposed to just keeping the structure smooth. So your lungs are a classic example of that. What you have inside your lungs is a huge amount of surface area because the structure in which the lungs are built creates not so much folding, but a much enlarged surface area. And we need that surface area in order to bring in the oxygen necessary to feed the cells, particularly, for example, when we're exercising. And any kind of disease process which attacks and reduces the surface area of your lung is going to compromise the rate of diffusion of oxygen into your blood, and that will show up in lower oxygen levels in your blood. So surface area plays a critical role, and we'll see that particular idea play out a little bit later on when we talk about ion channels. So let's move on, and let's talk about the fifth factor. So diffusion distance. Okay. So the greater the distance, the slower the rate of diffusion. The further you have to go, the slower it takes to get there. So let's turn to the lungs again as an example of where this can play out. So the respiratory membrane that I was talking about earlier, that has such a high volume of space inside the lungs, is very, very thin, extremely thin. And that's deliberate because we don't want to have a large distance between the capillaries that are capturing the oxygen and the oxygen that's in your lungs. So very, very thin in order for diffusion rates to happen very, very quickly. But what happens if your lungs, for one reason or another, begin to fill with fluid? Well, now that process of fluid building up in the lungs is going to create an additional layer of fluid on top of the respiratory membrane. And so now the distance that oxygen has to travel to get into the blood is increasing, completely dependent on how much fluid there is, and diffusion rates are dropping. And if that's significant enough, you will see a difference in the amount of oxygen being carried in the blood. So as fluid builds up in the lungs, you're going to get less and less oxygen being infused into the blood. So diffusion distance plays a very important role in all of this. So those are some of the factors that are going to influence diffusion. Let's, before we move forward and talk about the various types of diffusion and give you examples, let's take this moment and just kind of review what we've been talking about very quickly. And we'll do that with this little overhead here. So, as you can see, this is a very simple diagram that's going to show us a number of the things that we've just talked about. So, first of all, please notice that here we have a lot of sugar on this side, a lot of the gray balls on that side, many green balls, well, relatively, and more yellow balls than over here. So there's only one there, there's three there, there's three there, one there. So you can see we do have a concentration gradient, although this one is different than the others. So we have a concentration gradient, and we can take advantage of that. So, for example, this molecule here is using simple diffusion to cross the membrane. This molecule here is using a channel protein. This is channel-mediated facilitated diffusion. And this molecule here is using the other type of carrier-mediated facilitated diffusion. And we're moving that substance, which happens to be sugar, by changing the shape of this molecule and moving it across the membrane in that case. So this one is simple diffusion, channel, and then carrier-mediated movement of a molecule across the membrane. Again, the key thing here between this and this is an open pathway, and this one, literally, the protein has to change shape before it moves. And then over here we have active transport. And notice in this case, we're moving that green substance, whatever it is, against its concentration gradient. So it's highly concentrated over here, not very concentrated here. So we're going to take that and move it against that concentration gradient. In order to do that, we're going to need to work. We're going to have to use ATP. These are going down their concentration gradient, so no work is involved on the part of the cell. Although all of these systems have kinetic energy, and we're utilizing that kinetic energy to move things across the membrane in these passive transport mechanisms. The opposite happens here with active transport. Oakley dokley's. So let's talk about simple diffusion now. And let's try to understand what kinds of molecules are going to take advantage of this mechanism to get across the membrane of a cell. So we're looking at simple diffusion. And to remind ourselves very quickly, once again, what we're talking about, this is it here. We're looking at this process right here at the end. Okay, so simple diffusion. And we're asking ourselves what uses or what substances move by simple diffusion. Okay. And the answer to that is this. Number one, non-polar hydrophobic molecules. Nonpolar hydrophobic molecules. Okay. Remember that we're moving across a lipid bilayer. And by its very nature, lipids are hydrophobic. So anything that can mix with a lipid, another hydrophobic molecule, and as long as it's not polar, will cross that membrane and enter or leave the cell. Okay, so some examples are oxygen, carbon dioxide, nitrogen, fatty acids, steroids, fat-soluble, vitamins, okay, ammonia. I'm not giving you a complete list, enough to give you a sense of what we're talking about. So nonpolar hydrophobic molecules. All right. The other type of molecule is small polar molecules. And two good examples there is water and urea. These are very small molecules. They are polar, but they can cross the membrane. So the small polar molecules can also cross the membrane through this mechanism of simple diffusion. Okay? So that's simple diffusion. Let's look at facilitated diffusion. And of course, we have that in two different forms. So we're going to look at facilitated diffusion. And the first one is going to be channel-mediated diffusion. I'm kind of abbreviating this. My fingers are getting so tired, so I'm going to get kind of lazy. Channel-mediated diffusion. And of course, you know what we're talking about here. I could have thrown in the word facilitated, but I didn't. So that's my bad. Channel-mediated diffusion. And let's again remind ourselves what we're talking about here. So this is it right here. We're moving through some sort of pathway, unobstructed pathway. And for the most part, we're going to be talking about ions. Mostly ions are going to use this mechanism. So not necessarily all the time, but mostly ions is what we're looking at. And remember, one of the things that we said, about this carrier-mediated, the channel-mediated diffusion, or in fact carrier-mediated diffusion, is that each of these channels, each of these proteins are very specific as to what they carry. And they only carry a single type of molecule. All right, so channel-mediated diffusion. What about carrier-mediated diffusion? Okay, I should have added facilitated. I know, my bad again, but I'm not doing it. I'm getting lazy. So carrier-mediated diffusion is going to require a protein. It's going to be carried by a protein that changes shape. Let's remind ourselves what we're talking about. Yep, there it is right there. So when this molecule of sugar binds to that, it will change shape and move. In fact, I have a better example of that. So here it is. There we go. And right side up. And here we go. Okay. So move this up a little bit. So glucose binds to the molecule. By binding to the molecule, it causes that protein to change shape. The change in shape of the protein moves that glucose across the membrane. Again, we're going down the concentration gradient from high concentration to low concentration. Okay. Now, one of the interesting things that happens is as soon as the glucose comes in, we're going to attach a phosphate group to that, and that is going to be done by an enzyme. Here you can see the enzyme hexokinase is the enzyme that facilitates that reaction, and we're going to set that molecule up now to add or make larger molecules out of it. We can start building on this molecule by adding that phosphate group there to it, and that you'll learn more about later on. So that's we've got a transporter protein carrying and changing shape the amino of the substances. So what kinds of things are generally going to move are going to be sugars, particularly glucose, some vitamins, amino acids, now, there is a little bit of a wrinkle here with carrier-mediated facilitated diffusion in that some of those transport proteins, so some transport proteins are gated. And we have to talk about that a little bit. So, and generally, though not all of them, what we're talking about are gated ion channels. So, and these are open or closed. They're regulated by two possible stimuli. One is electrical gradient, and the other, and is less common, is chemical gradient. Okay. So let's look into that a little bit and show you what that's all about. So first of all, I'm going to use a picture from the textbook just to show you what we mean by gated, and then I'll explain this regulated part of our notes. So gated ion channels, they're a part of the carrier-mediated diffusion. They're a special type of protein that has a gate attached obviously, as the name implies. So here's a good example of what we're talking about. So I've got my membrane, and here's the carrier protein, and there is the gate. In this case, the gate is open. In this case, the gate is closed. So what causes that to open or close? And that can be two things, as we mentioned. Either a difference in the concentration of charges across the membrane, or a difference in concentration of some chemical across the membrane. Now, the difference in electrical charges is much more common. And in fact, you're going to run into this and have to understand this in some detail when you get to 40B and look at an action potential in a neuron. So let me draw a very quick picture to help us understand what we mean by this electrical charge gradient across the membrane, either opening or closing one of these gated ion channels. So let's do that right now. And I'm going to give you two scenarios. So and then that's the first scenario. And then the second scenario is over here. And what we want to do is we want to so we've got these channels, and let's go in and kind of give this some shape here. Okay. Okay. So there they are. And let's go ahead and add a gate. So in this case, it's closed. In this case, it's open. And we're going to go in and we're going to highlight that gate. And just because I can do it, I'm going to kind of get my artistic mode happening here. Okay. So this is closed. This is open. And why is that one closed and why that one is open is the question that we have. And the answer to that is, in this case, we're going to stipulate the following. We're going to say that if we were able to get a little voltmeter, and we actually have these, people have built these things, and they have these little voltage meters that can measure the difference in voltage across the membrane. And typically, for a typical cell in your body, the difference is going to be somewhere about 90 millivolts, in that neighborhood. And that's your, if we can say this, that's your homeostatic condition, is a difference in charge across the membrane of 90 millivolts. Now, why did I make it negative? Because remember, the outside of the cell tends to be negative, and positive, and the inside of the cell tends to be negative. So this is intracellular, and this is extracellular. Okay. And so we attach the minus signs because we're always using the inside of the cell as our frame of reference. And since that's a negative condition, we attach the negative sign to the difference in voltage. So a difference of 90 millivolts across the membrane. And in that condition, that gated ion channel is in the closed condition. Okay? Now let's say something happens and we reduce that difference to, I don't know, let's make it, I'm just going to make up a number here, 30 millivolts. Conditions change, and we now have, instead of a 90 millivolt difference, we have a 30 millivolt difference across the membrane. And as a result of that, this protein has noticed, or read, if you will, it's acting like a little voltage meter. And it has read that difference, and this is the trigger point right here. When it reads that difference, this will pop open. And then, of course, ions will move. So it's open because that stimulus causes it to open. It's closed when it's experiencing this condition. Okay, so that's an example of voltage-gated ion channels, and it's an example of how they operate and what they do. Okay. All right. So we've talked about simple diffusion. We've talked about facilitated diffusion. That leaves only one other passive process to talk about, and that's going to be osmosis. So we're going to discuss osmosis. And this is going to be the net movement of a solvent, which for us is going to be water, H2O, okay, through a selectively permeable membrane from a region of high concentration to a region of low concentration. And so here we have a definition that sounds very, very similar to diffusion. There are two important differences. Number one, we're talking about the solvent, not the solutes moving. And the reason that the solvent is moving is because we're preventing the solids from moving because we have this selectively permeable membrane. So it's a membrane which only certain things can cross. That's the idea of selectivity. It's permeable, but not to everything. It's selective as to what is moving across the membrane. Okay? So, when we have this process going on, we are going to experience a couple of conditions, or this movement, I should say, is the result of three possibilities. Okay? So, I have already laid out in somewhat more detail than I am going to in this lecture in the lab the idea of osmosis. And so I want you to reference that in relationship to what we're talking about here. So I'm not going to go through all that detail again. I'm going to kind of highlight a couple of things that we've already been exposed to, and I'm going to introduce another concept which I did not talk about in the lab. So we talked about tonicity. And tonicity is the idea that we can have different types of solutions. So, for example, we can have an isotonic solution. And that means we have the same concentration of ions outside the cell as we have in the inside of the cell. So same concentration, isotonic. We can have hypotonic solution, in which case the concentration of solutes on the outside of the cell is less than in the inside of the cell. Or we can have a hypertonic solution. And in that solution, the concentration of solutes on the outside of the cell is greater than on the inside of the cell. And each one of these conditions has implications for the conditions of that cell. So in isotonic conditions, you're going to have a normal shape. So isotonic means normal shaped cells. So that is something that your body works very, very hard to maintain, is an isotonic condition around the cells. So normal shaped cells, if you have normal shaped cells, you're going to have a normal function. Because remember the old adage, form follows function. And your cells in your body are shaped in certain ways because they're performing certain functions. Okay. So let me turn on a little bit more light. So we're getting dark here. And that might help. All right. Hopefully it does. So isotonic is what we're aiming for because that gives us our normal shaped cells and we get a normal functioning cell from that. What happens with a hypotonic solution? Well, in this case, what you're going to get is hemolysis, right? That's if you're talking about a red blood cell. If you're talking about any other cell, it's just simply going to lyse. So lysis. That is, the cell's going to break apart. Water is going to flow into that cell and eventually will break the cell or explode the cell, swell the cell to the point of it losing its integrity. That's a hypotonic solution. Obviously not something that you really want to have happen. And the last scenario is hypertonic. And in the hypertonic solution, what you're going to get is crenation. That is, water will flow out of the cell. Again, we're losing the shape. And because we're losing the shape, we're losing the function of that cell. So we got three possibilities, only one of which is good for us. And that's the isotonic solution. Okay. Now I want to talk about osmotic pressure. And this is something I did not mention in the lab. So this is an important concept because that's how we're going to measure where water is going to flow. So let's give ourselves a definition of osmotic pressure. So osmotic pressure is proportional to the concentration of solutes that cannot pass across the membrane. Or another way to say the same thing is higher solute concentration or I should say maybe the higher, let's do it that way, the higher solute concentration, the higher the osmotic pressure. So the higher the solute concentration, the higher the osmotic pressure. Let's go ahead and illustrate that to try to understand what we're talking about. So I've got two conditions. And we're going to place in that a selectively permeable membrane. So that's our selectively permeable membrane. And then we're going to have some, of course, we're going to have a fluid in there, so there's going to be some water. Okay. And now I'm going to go ahead and I'm going to bring in some, let's see something, yeah, there you go. I'm going to so there is that scenario. And then here is another scenario. So kind of two different scenarios. And the pink is, of course, the solutes. Let's take example A first. Now the question is, I have area 1 and area 2, which one of these has the higher osmotic pressure? The higher osmotic pressure. So let's go back to our statement. The higher the solute concentration, the higher the osmotic pressure. So clearly this has a higher osmotic pressure than that. So this has the highest osmotic pressure. What about in scenario B? Well, and this is it's not so dramatic as this scenario, but it's still, this has a higher solute concentration than that side, so this has the higher osmotic pressure. Now ask yourself, where is water going? So this is hypertonic compared to that. This is hypertonic, that's hypotonic. This is hypertonic, that's hypotonic. So water is going to move from high concentration to low concentration. Water is moving in this direction, is it not? Say, yes, teacher, it is. And water is moving in this direction. And the reason I illustrate this is because students often get confused and they hear that higher osmotic pressure, and they hear that word pressure, and they think they're pushing water out of a region across the membrane. That's the exact opposite of what is actually happening. So when you have a body of fluid that has a higher osmotic pressure than another body of fluid, water is literally moving into that area that has the higher osmotic pressure. And the best way to think about this is if you were a person standing right here, and you got a little happy face there. If you're standing right there and you're feeling that water come in against your body as it flows from this region into this region, that's a pressure that you're experiencing. The pressure of the water moving into this space. That's the osmotic pressure. That's a higher osmotic pressure, okay? This has an osmotic pressure too because it has some solutes in it, but does not have the same as this. Has the higher osmotic pressure. This has some, this has more. So water is going to move from this area to that area, okay? So really it's very disconcerting, this idea of higher osmotic pressure, because it's very easy to translate that term into some kind of water is being pushed out of an area into another area. So if something has a higher osmotic pressure, there's more pressure against the water to move out, when in actuality it's the exact opposite of what is happening. Okay. All right, so let's take a look and re-emphasize what all of this means by way of some simple pictures. And we have seen this, and we've certainly talked about this in the lab. So here is my conditions. I have an isotonic solution. I have a normal shaped cell. That's exactly what you want. 1, 2, 3, 4. 1, 2, 3, 4. Okay. I have a hypotonic solution. I only have 2 particles here. I have 4 there. So water is moving in, and we get hemolysis. Now remember, again, we're talking about red blood cells, so I can use this terminology, hemolysis or hemolysis. If this were not a red blood cell, I could only use that term right there to describe what's happening to that cell. So this is a normal shaped cell, normal function. This one clearly is not going to function because it will no longer exist. And the last example is the hypertonic. So I've got 6 out here, only 4 here. Water's flowing out, the cell shrinks, it's losing its shape. Remember, this is what we wanted. This is the shape we wanted. That's the normal shape. That's the way it's designed to be functional. If I change that shape in either of these conditions that's happening, I'm not going to get a functional cell. All right, so that leaves active transport. And So we're almost done here, folks, so hang in there. I'm trying to. It's late, and this is literally the third time I've done this because the first 2 times I made mistakes, so I had to scrap and start all over. But that's okay. Things like this happen. We're talking about active transport now. Active transport, of course, to remind ourselves, we're going to use ATP. We're going to move against the concentration gradient. Okay. Some kinds of things that will often be moved in this direction are amino acids, sugars. There's a variety of things. Just giving you some quick examples. All right. So we have primary active transport. That was one of the ones we identified that falls under this rubric of active transport. And let's go way back to our original drawing, if I can pull that up real quick like. So active transport is illustrated quite clearly right here. So I'm going to move this green ball from this side to that side using ATP against this concentration gradient. So this is what I'm interested in, moving that thing right there, whatever that is, across the membrane in this direction. And then I move it by using energy. That's ATP. So primary active transport is I'm going to move a substance, and I'm going to use this word directly across the membrane using ATP. Okay. Why did I use the word directly? You'll see in a moment when I talk about secondary active transport. Secondary active transport. So let's take a look at this. And believe me, this really gives students fits. So I'm going to try very hard to make a distinction between secondary active transport and primary active transport. So secondary active transport, let me go ahead and give you an example of this by way of a picture. This is actually something that happens in the kidneys. So what I'm showing you is something you will see again when you study the urinary system. So I'm going to use a clean page. I'm going to draw a picture here to illustrate what we call secondary active transport. I think it's pretty clear, hopefully it is, for most students, what primary active transport, we're moving it directly using ATP. But what's this secondary active transport? What's that all about? So let's do that. And we're going to take our time and draw a nice little picture here. So get your artiste hat out. Okay. So we're going to fill this in. Okay. And let's bring in some color, shall we? Yeah, I'm going to bring in some color so we can make this pop a little bit. Okay. And we're going to label these in just a second, folks, so hang in there. Bring this. Okay, I can get away with that. Okay, so notice that I left a space there. We're going to need that space. So I'm going to bring in something here. Okay. And I've got something here. Okay. And then I've got something here. We're going to go ahead and bring in some more color because we're really digging this. Okay. Nice. I think I'm getting a little bit loopy here. Okay. All right, so let's label things so we know what we're talking about here. So this is going to be the lumen of the kidney tubule. And that may mean absolutely nothing to you and not be very instructive at all. But that's going to be the lumen of the kidney tubule. And then this will be the cells. Let's put this up here. This will be the cell making the wall of the tubule. So the wall of the tubule is made up of these cells. I've exaggerated the size of the cell, and I've only shown one. And this is going to be my blood capillary here. Okay. And I'm going to need a sugar molecule. Oh, there's my sugar molecule. Okay. And I'm going to go ahead and let's do a little green action on that. Okay. And I'm also going to have some sodium here. In fact, I'm going to have 2 molecules of sodium. Okay. All right. So we've set the stage, and let me just give you a little background so this makes sense. This is my kidney tubule, and what has happened before this stage that we're drawing here is we have filtered the blood into the tubule. So the first part of the nephron, which is the tubule is part of, we filter the blood, and we filter everything except for proteins, basically. So even glucose, amino acids, everything we want to keep, plus everything we don't want to keep, all the waste, they're all filtered together. Now they're inside the tubule. And our problem is we want to recapture the things we need, like glucose, and leave the waste behind. And this is one of the mechanisms in which that happens. So over here, I'm going to use a little bit of energy, and I'm going to pump sodium out, and eventually it'll get back into the blood. But I'm going to use up ATP right here. So I'm going to take ATP, and I'm going to convert it to ADP. So adenosine triphosphate to adenosine diphosphate. I'm using up ATP. I'm working to move sodium out of the cell. So basically, this concentration of sodium inside the cell is reduced because I work so hard using energy to pump the sodium out. Okay? So that creates a concentration gradient between the inside of the cell and the tubule. So if I look at this, that concentration is high compared to this because I've done this work right here. And I've done that work using energy. Okay. Now I've created a certain condition, low concentration of sodium. High concentration, what's going to happen? Sodium's going to want to come into the cell because we have a difference in concentration. And we're going to say sodium, oh yeah, you can do that. Come on in. The water's fine. But we're going to tell the sodium, in order to come in, you're going to have to bring in with you glucose. It's going to drag glucose along with it. It's moving down its concentration gradient, and in so doing, it's doing work for us by bringing that glucose into the cell. So now the glucose is in the cell, and then we're going to provide a mechanism for that glucose to end up back into the blood. Okay. So we're just providing, this is facilitated transport here. This is probably carrier-mediated transport. What we're interested in is capturing that molecule there. This is our target. We want more of that back into, we've already filtered it, that's why it's here, but we want to bring it back into the blood. And the mechanism we're doing that is we're using energy here to create a low concentration of sodium inside the cell so that the sodium will want to come through and drag with it that molecule of glucose. This is secondary active transport. We create, by using energy here, the conditions to grab and move the things that we want to move over here. These two things are connected together. These two things are connected together. Don't ever disconnect those two things. If you disconnect that from this, then you are going to get confused with active transport, because if you simply look at that, you're saying, oh, that's active transport. I don't understand secondary active transport. No, you don't, because you forgot to connect these two things together. That energy use is connected to this movement. They're linked together. And since they're linked together, it's secondary active transport. Here, we're using the energy, and secondarily, we take advantage of that use of energy to drag this molecule into the cell and recapture it into the circulatory system. So secondary active transport. And as again, I'm going to repeat this because it's so crucial to your understanding and so you don't get confused. Always connect these two events. If this event doesn't happen, that event doesn't happen. They are connected as if they were chained together. Don't ever do this. Then you're going to get confused. Then it's active transport. And then you're saying, what's going on? No. These two things are connected together. And don't disconnect them in your mind or you will get confused. Okay? All right. Perfectamundo. Let's move on. That's secondary active transport. Secondary active transport. Now, with secondary active transport, there's one other wrinkle. There are symporters and antiporters. So before I explain this, I want you to think a little bit and ask yourselves, now that you've been exposed to the idea of how we put together words in science to create meaning out of another word, anti means against, right? Sim means similar, same, or with, sympathetic, same, okay? And what we're talking about, and I have an example of this, and here we are. So let's take a look. No, that's not what I wanted. And, all right. Here we are. Let's do this. I am getting loopy. Okay. So here are the examples. Antiporters. What's going to happen is the molecule we're interested in is going in opposite directions. So we move sodium out, we create a concentration gradient with sodium, and in this case, sodium moves in as another molecule moves out. So calcium is the one we're interested in, that's what we want to move in the first place. We're using a difference in concentration that we've established through secondary active transport to move calcium, and in this case, they're going in opposite directions. Hydrogen, the same thing, going in opposite directions. Ooh, there we go. Here, we're going in the same direction. Symporters. So it's the same principle. We're moving things in one direction or another, but in this case, we're going in the same direction. The thing we're interested in, glucose. The thing we're interested in is amino acids. And we're moving them in the same direction as the sodium. And now I bet somewhere out there in the ether, a student is saying, hey, wait a minute, look at all that. We've got a lot of sodium here, and we're moving sodium down its concentration gradient, so isn't that diffusion? And my answer is no. Why? Because I told you to never disconnect these two events from each other. So the reason why sodium is in such a low concentration here inside the cell is because we worked to produce that difference in concentration. And then we took advantage of that to move something. And when we move that something, it either can move in the same direction as sodium or in the opposite direction of sodium. If it's moving in the opposite direction, that's an antiporter. And a symporter is they're moving in the same direction. Okay? Now my question to you, ladies and gentlemen, is which one is this? What example did I show you here? Is that an antiporter or a symporter? And the answer is, you're right. That is a symporter, moving in the same direction. And in fact, it's exactly the example that they gave you in this image here. There's my glucose, there's my sodium, low concentration on the inside, high concentration. This would be the tubular filtrate, the material that's in the tubule of the kidney, and we're moving it in this direction. Recapturing the glucose, we do that with amino acids as well. But we don't have to go in the same direction. We can go in the opposite direction as well. Okay, symporters or antiporters. So coming back then, a very simple definition. Symporters move substances together in the same direction. Antiporters move substances in the opposite direction. Okay. Woohoo! Almost done. So what's next? What is next? What's next is to talk about vascular transport. So let's do that. Vascular transport. And I'm not going to spend a lot of time here. I'm going to show you the three different subsets of vascular transport and just very quickly go over those. We don't spend a lot of time with that. The last one, phagocytosis, you will run into again when you do the immune system. But remember, when we listed those, they are number one, endocytosis. Number two, exocytosis. And number three, phagocytosis. Okay. And let's go ahead and just simply look at a couple of examples. So this first one is going to be endocytosis. And again, you said looking at the words and thinking about them a little bit. There you go. Endo meaning incoming into the cell. Cytosis referring to cell, into cell, out of cell, and eating. Not, it says literally eating cell, but that's not what we're talking about necessarily. But anyway, talk about that. Okay, here we go. All right, endocytosis. Here's what we want to capture. Don't worry about the receptor and all that kind of stuff. That's not really something we need to pay attention to. The point that we're doing, or the making, is that we're capturing something on the outside of the cell. In this case, we have receptors that bind to that. And then we create a vesicle and pinch that off and bring it into the cell. And then we process whatever it is that's come in, and whatever that happens to be. And there's many, many examples. We don't have to worry about that part of it. The point that I'm making is endo, we capture something by creating the vesicle and bringing it into the cell. That requires energy. This process is an energy, requires energy for the cell to do. It's work for the cell. So it needs to use up ATP. Okay. The opposite can happen. As a matter of fact, I don't have really a good example of that, so I'm going to go back to this one. I'm just going to pretend that we're going in the opposite direction. Let's say we're making this inside the cell and we want to extrude it out to the outside of the cell. We'll form a vesicle, contain the material that we want to get rid of, and then that vesicle will merge with the membrane and we move things out. That's exocytosis. So endo coming in, exo going out. Very simple. Phagocytosis is the process of bringing in, as you can see, larger things such as a microbe. We capture a microbe, bring it into the cell. Instead of processing in some way, we're going to digest it. We're going to break it down and digest it to destroy it. And that would be phagocytosis. So that's an example of phagocytosis. So larger particles, quite often things like bacteria, or cellular debris. So if you've injured yourself and you had dead tissue in an area of injury, that dead tissue can be gotten rid of through phagocytosis. All right, ladies and gentlemen, we are finished with that part of the chapter. I have one more lecture, and in that lecture we're going to talk about cellular organelles, and it's going to be so much fun, you're going to die of funniness. And I'm going to get out of here before I make any more ridiculous comparisons.